All right, folks. So chapter 25, I'm going to post this before some of it before spring break. I know that it's not due yet, but in case some of you want to get ahead. So this is metabolism and energetics, and it's so you just finished digestive system, and we talked about all the hormonal and neuronal control of digesting food and absorbing it. So what do we do with these nutrients after they're in our body? Well, first of all, let me remind you that metabolism is the sum of breakdown. Whoops, didn't mean to do that. Metabolism is the sum of breakdown, which is catabolism, and buildup, which is anabolism. Now, anabolism is endergonic and requires energy, and that energy comes from ATP. ATP drives the anabolic reactions. Now, catabolism is exergonic for the most part, and this gives you ATP when you break it down. So this is a cycle, and really there's an ATP cycle here, where anabolism uses ATP because they are endergonic reactions, and catabolism generates ATP. So this is our nutrient pool. Concept of a nutrient pool is our monomers, our amino acids and simple sugars, and some of our lipids, which aren't built on the monomer polymer system, as you may recall from Bio 203. But amino acids are the monomers of polypeptides or proteins, and simple sugars are the monomers of polysaccharides or carbohydrates. Okay, so this is an overview. We're going to start talking about some of the details of this. This is, again, more of an overview. It's saying from your nutrient pool, your fatty acids could go through an anabolic pathway and be converted to triglycerides, or they could go through a catabolic pathway, be broken down by what we call beta oxidation, which we'll cover, and you generate ATP. So they can go either way, and really the control is the key thing. I don't want you to memorize all of these metabolic pathways by any means. But I do want you to understand the detailed control mechanisms of how and why a cell does what it does. Likewise, glucose. It's a monomers of polysaccharides. In animals, that's glycogen. In plants, that's starch. So the glucose could go through anabolic pathways and be converted to glycogen, or it could go through catabolic pathways and be burnt into CO2 and water, and you generate ATP. Amino acids. Amino acids can go through anabolic pathways and be converted into proteins, or they could go through catabolic pathways and you could burn them and generate ATP. Now, typically speaking, amino acids, there are some couple hormones that uh, preserve the amino acids. They keep them from being funneled into catabolic pathways. But that's not perfect. Amino acids are indeed funneled into catabolic pathways, just like glucose and fatty acids are. But after a certain period of time of being calorically deficient, meaning not having enough glucose or fatty acids in your diet, you do release some hormones that save the amino acids. So it really takes a severe starvation to burn your proteins to a great deal. I mean, our body tries to save our muscles and our other proteins like collagen and other lean body mass proteins. We do burn them a little, but for the most part, we try not to.
Now that you've learned about different catabolic and anabolic processes of microorganisms, let's see how these fit together into a complete picture. Proteins can be broken down by protease enzymes into amino acids, which can be further broken down through the glycolysis and Krebs cycle pathways. Nucleic acids can be broken down by nuclease enzymes into nucleotides that can also enter the glycolysis pathway. Polysaccharides and other sugars can be converted into glucose and further broken down through glycolysis and the Krebs cycle. Lipase enzymes break lipids into glycerol, which enters the glycolytic pathway, and into fatty acids, which enter just prior to the Krebs cycle. Fermentation is also considered catabolism since it converts pyruvic acid into other 2 and 3 carbon molecules, as well as carbon dioxide. Recall that anabolism is synthesis of complex molecules from simple molecules. Essentially, anabolism is the reverse of catabolism. Note that synthesis of proteins, nucleic acids, polysaccharides, and lipids is the reverse of their catabolism. The pentose phosphate pathway uses an intermediate from glycolysis to produce 5 carbon sugars called pentoses. These pentoses are then useful in synthesis of nucleotides. Photosynthesis is primarily an anabolic reaction, using light as an energy source to build complex molecules like glucose from carbon dioxide.
All right, we don't have to worry about photosynthesis, folks. It's just in here. Let me go back to these catabolic pathways. He said something I don't agree with. I understand why he's saying it. I don't completely disagree in his context, but I just want to be clear about something. These yellow arrows are catabolic pathways. You can see the key right down here. Whoops. These yellow arrows are catabolic pathways. There's the key. And I don't want you to memorize, I definitely don't want you to memorize any intermediates. That means I don't want you to memorize any of the intermediates between glucose and pyruvic acid. It would be nice if you knew that burning glucose to 2 pyruvic acids is called glycolysis. And it would be nice if you knew that taking pyruvic acid and converting it to glucose is called gluconeogenesis. It would also be nice if you knew that lipids are funneled into some of the main pathways. In essence, the lipids are funneled into the Krebs cycle. The glycerol part of the lipid is funneled into glycolysis. It would be nice if you understood that protein metabolism, the amino acids can enter in different places of the catabolic pathways. For example, some amino acids enter as pyruvic acid, some enter as acetyl-CoA, some enter directly into the Krebs cycle. So you're not going to have to memorize any of the intermediates of these pathways, but an overview about how we burn proteins, how we burn polysaccharides and lipids is nice, just as an overview. Now, he said something right around in here. He said it after the yellow arrows came up. He said that anabolic pathways like gluconeogenesis and fatty acid synthesis and nucleic acid synthesis, he said these were essentially the reverse of catabolic pathways. Well, in one sense, he's not incorrect. But I will tell you that there are significant deviations. For example, we're going to look at gluconeogenesis. We're going to look at gluconeogenesis, and you're going to see that it is not the exact reverse of glycolysis. It indeed isn't. There's quite a major deviation. I mean, many of the steps are simply reversal reactions. Many of them are, but there's a major deviation, and there's a reason for that deviation. So yeah, I want you to think that, okay, these anabolic pathways like gluconeogenesis and protein synthesis and lipid synthesis, etc., they're dehydration synthesis reactions, many of them. Not all of them, by the way. And they're kind of the reverse of the hydrolytic reactions of catabolism. But they're not exact reversals. They certainly are not exact reversals. Okay, so that's 25.1, and that's actually the easy part of this chapter. 25.2 gets more difficult.